Now we are discussing the topic reproduction. So here we are discussing the topic that is a carpel or megasporophyll. Carpel or megasporophyll. So when we are dealing with the topic that is a carpel or megasporophyll, we all know that carpel is a structural and functional part of gynoecium. It is a structural and functional part of gynoecium. When we talk about the carpel, it is divided into three parts. It contains stigma, style and ovary. Okay. When we see the structure, stigma, style and ovary structure, this is stigma, style, and this swollen part is known as ovary. This is the basic structure. Now we are dealing with the structure of ovule or megasporangium. Structure of ovule or megasporangium. So when we deal with the structure of ovule and megasporangium, first we have to deal about the ovule which is present inside the ovary. Now ovule is an outgrowth of placenta and ovule is outgrowth of placenta. Each ovule is connected to its placenta by a stalk called funicle. Let me first draw the diagram. Okay. Now we are discussing the diagram first. First we are discussing about the ovule. This is the structure of ovule which is the outgrowth of placenta. Each ovule is connected to its placenta by a stalk called funicle. So when we talk about funicle, this is funicle. Next, the point of association of the funicle with the main body of the ovule is called hilum. So when we talk about the hilum, is known as hilum, where the funicle attached to the ovule. The main body of ovule is called nucellus. So this is nucellus. This nucellus is made up of a parenchymal tissue. Then each ovule has one or two protective envelopes which are known as integuments. So these are the integuments. Integuments encircle the ovule except at the tip where a small opening is present which is known as micropylar end. Which is known as micropylar end. This is micropylar end. And opposite to the micropylar end, this end is known as chalazal end. Chalazal, which represent the basal part of ovule. So this is the structure of ovule or megasporangium. Now we are dealing the topic that is megasporogenesis. Now next topic is megasporogenesis. So when we deal that is the topic megasporogenesis, it means the formation of megaspores from the megaspores mother cell. In simple form, we will write the process in which MMC, that is the megaspore mother cell, give rise to megaspores. And this process is known as megasporogenesis. In this, ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. Basically, megaspore mother cell is mostly present in that micropylar end. You all know that this is the micropylar end. This structure, this is the micropylar end, and here in the micropylar end, megaspore mother cells are present, which give rise to the formation of megaspore. These megaspore mother cells basically undergo meiotic divisions, and these meiotic divisions ultimately result in the formation of 4 megaspores. Let's see the structure. When we see the structure, suppose this is structure. This is the structure. Here, this is nucellus, and here the microspore mother cell is present, which we say megaspore mother cell. This mother cell undergo meiosis, and it forms first. What happens there in megasporangium? Okay, what happens there? But occur here? Let's see. First we will explain the diagram. Here two cells are formed. That is a microspore diad is formed. This is known as megaspore diad. And then further this is meiosis first, and then it forms meiosis second. And here what happens? Spore, यानी कि megaspore tetrads are formed. Megaspore. Okay. Now we again discuss this topic, that what happens here in the megasporogenesis. One of the cell of nucellus differentiate to form megaspore mother cells. It differentiate and form megaspore mother cells. MMC means megaspore mother cell. This megaspore mother cells, it undergo meiosis and form two cell stage, that is known as megaspore dyad. Megaspore dyad is formed. After this megaspore dyad, again it undergoes meiosis and it forms megaspore tetrad. Megaspore. Okay. After the formation of megaspore tetrad, in this tetrad means 4 cells. In this out of these 3 cells, tetrad, 3 cells will undergo degeneration, will degenerate, and one remain functional. This functional megaspore. Now what happens? Now we are dealing with the functional megaspore. This functional megaspore again undergo nuclear division, okay, and forms two nucleus. These two nucleus divide and form 4 nucleus. This again called division, and it forms 4 nucleus. This 4 nucleus again undergo division, and it forms 8 nucleus. Okay? And these 8 nucleus arrange themselves. What happens here? Let me see. First, we have seen that there is only a single. This is a single megaspore mother cell. Simple. This single megaspore mother cell undergo 2 cell division. First cell is there, second cell is there. Then this again undergoes cell division. These 2 cells again undergo cell division and form 4 nucleus. Here 2 nucleus. Now what happens? This again undergo and form 8 nucleus. 4 above. This is the structure up till here. Keep 8 nucleus up. Now these 8 nucleus will arrange themselves in a definite pattern. Now this 8 nucleus, how they arrange themselves in a definite pattern? Let's see. They arrange them in a definite pattern where these are the antibodies and 3 cells. Here 2 synergids. These are 2 synergids. Here a H cell, and in the center 2 polar nuclei. Okay, this is the structure that is formed. Okay, now we are discussing the topic again. What happens? These 8 nucleus will arrange themselves in a definite pattern. 3 will move towards the Schlesinger line. These are antibodies which moves to the Schlesinger line. This is micropylar end. Here we discussed that in this 8 nucleus will arrange themselves in a definite pattern. 3 will move towards the Schlesinger end and will form antipodal cells. Other 3 will move towards the micropylar end. The middle cell is known as X cell. This is known as X cell. And these two are known as synergids. These two are known as synergids. That cell is surrounded by 2 synergids. The work of this synergids is to secrete hormone and attract pollen grains. The remaining 2 nuclei remain in the center of the embryo sac and form a single cell which is known as polar nuclei. This is known as polar nuclei. This is basically 2 in nature. Now, below the micropylar end, Filiform apparatus is present that help in guiding the pollen tube. In synergids, filiform apparatus is there. Filiform apparatus is there which guide the pollen tubes, which guide the pollen nuclei and guide the pollen tube. This is complete female gametophyte which is known as 8 nucleated stage. This stage is known as 8 nucleated stage or 7 cell stage or 7 cell stage. Because why we are using the word 7? Because two nucleus are fused together and form a polar nuclei or a polar cell. Next, we are dealing with the topic that is the pollination.